Good morning children, I am Sadhya. Now I am going to teach you the mechanism of DNA replication. As you know, energetically the replication process is very expensive or highly expensive. So, a number of enzymes act as catalyst or that can catalyze the DNA replication. So, before getting to the details of mechanism of DNA replication, we have to see which are the enzymes involved in this process. Okay, let's see which are the enzymes involved in the DNA replication process. The first enzyme is helicase, DNA helicase. Okay, so as I told you, in the two strands of DNA, both the strands act as template. Template strand means the strand which is capable to synthesize new strand. So both the strands act as template strand to form new strands. So before starting replication, these two strands need to be separated. That separation is accomplished by this enzyme helicase. For example, if this is the two strands, in between hydrogen bonds, nitrogen bases are there which is linked by hydrogen bonds. Okay, so these hydrogen bonds are broken by this helicase enzyme to get separate. Okay, for the unwinding of the two strands, this helicase can break the hydrogen bond between the nitrogen bases and that DNA strands get separate. Okay, so that is a function of helicase. So the function of helicase is unwinding of DNA strand. Unwinding of DNA strand for replication. Okay, and the second enzyme is primase. The enzyme primase. So the function of this primase is to add RNA primer. To add RNA primer to the DNA template. DNA template strand. Okay, so this primase can add an RNA primer. So what is RNA primer? RNA primer is a short stretch of RNA molecule. It consists of 10 to 16 nucleotides only. RNA nucleotides only. So it is a short stretch of RNA molecule. Okay, so this primer can add on the template strand to accomplish the further polymerization of DNA replication by another enzyme that I will teach you. Anyway, the function of primase is to add an RNA primer to the DNA template. Okay, and RNA primer is a short stretch of RNA molecule which consists of 10 to 16 nucleotides that can be added to the DNA template for replication. That I will show you. And next, the third one is DNA polymerase enzyme. DNA polymerase enzyme. And this DNA polymerase enzyme, it is the most important enzyme in the DNA replication. It is also known as DNA dependent DNA polymerase enzyme. Okay, and it helps for the, or it catalyzes the polymerization of deoxyribonucleotides. That means the elongation or the extension of the nucleotides. The new nitrogen bases are added through this polymerization process. So, the extension or the synthesis of new strand is accomplished by this enzyme, DNA dependent DNA polymerase enzyme. This is the important enzyme in the DNA replication process. And the fourth one is DNA ligase. Ligase enzyme. This ligase enzyme used used to connect the DNA fragments or used to join the DNA fragments. To join newly formed DNA fragments. In some cases, some fragments are formed in the discontinuous strands that I will teach you, and this ligase helps to join the DNA fragments in the newly formed DNA. Okay, newly formed DNA. Some discontinuous strands are there or lagging strands are there, and in 5' or 5' 3' template strand, and that fragments can be joined together by this enzyme ligase. Is it clear about the enzymes involved in this replication process? Now we can see the process of replication. Next we can see the processes of DNA replication. The first step in the replication is initiation. The first step is initiation. So in initiation, there is an unwinding of or separation of DNA strand. So it is done by the enzyme helicase. So here in the two strands, both the nitrogen bases are linked by hydrogen bonds, 2 or 3 hydrogen bonds, and these hydrogen bonds can break by this helicase enzyme. Okay, so this helicase enzyme can break the hydrogen bond between the two strands and the strand can be separated. That is the first step. And the second step is elongation. Elongation means the synthesis of new strand. Synthesis of new strand. That is the second step. Synthesis of new strand, and the synthesis of new strand is done by DNA dependent DNA polymerase enzyme. DNA dependent DNA polymerase enzyme. Okay, next we can see the processes in detail. Here, as I told you, the enzyme helicase separate. Okay, so this is the helicase enzyme. Here we can see helicase enzyme. They separate the two strands. It is in a longitudinal manner. So there is a Y-shaped structure formed, and that structure is called a replication fork. Replication fork. So replication fork is formed by the helicase enzyme by separating the two strands, and these two strands act as template strands. So here the two strands can act as template strands. So this is the replication fork made by the helicase enzyme. This is the helicase enzyme. Okay. And next we can see here, suppose this is a single strand, this is another strand. So the enzyme helicase separate these two strands, and then if this is the starting point of replication, this point is called origin of replication. Origin of replication. So the particular point at which replication starts on the template strand is called the origin of replication or origin. Okay. So in prokaryotes only a single ori can be seen, but in eukaryotes many ori can be seen. So here this ori, in the region of ori, an RNA primer is added by the primase enzyme. I told you RNA primer is a short stretch of nucleotides. It's an RNA molecule. It consists of a short stretch of nucleotides. This RNA primer helps the DNA polymerase enzyme for polymerization. Okay, so after that, this is the primer. After that, the DNA polymerase enzyme can do the elongation process. Elongation process means adding of nitrogen formation of a new strand. And this must be complementary to the template strand. So this is the template strand. And one more thing I want to tell you, the DNA polymerase enzyme can add or do polymerization only in 5' 3' direction. So the polymerization can be done by the DNA polymerase enzyme only in 5' 3' direction. So here this is the 5' 3' newly formed strand. Then what may be the polarity of the template strand? This is the polarity of the template strand. Am I right? Suppose, I'll tell you another example. Suppose the primer is given here. This is the ori. Okay, the primer is given here. So this is not the ori. This is the ori. So the primer is added here by the primase enzyme and the DNA polymerase enzyme do the elongation in this direction. That must be 5' 3' direction. Then what may be the template strand polarity? This must be 3', 5'. Is it clear? Okay. So here, what is the polarity of this one? 5', 3'. This is another template strand. Second strand. Okay. And one that template strand. So how this template strand form a new strand? As I told you, the DNA polymerase enzyme can do the polymerization only in 5', 3' direction. So here, this direction they can do the polymerization. Can it do the same direction in this template strand? No, because this is 5'. They must be anti-parallel. So they need to start the polymerization in opposite direction. Okay. So here the primer want to be added by the primase. Okay. Then only a strand is formed in 5', 3' direction. So fragments, this is in opposite direction. So due to this, discontinuous strand will be formed by the DNA polymerase enzyme. Okay. So here this strand is called a leading strand. This is the leading strand and this is the lagging strand. So this is the lagging strand which consists of fragments. Is it clear? So here the leading strand is a newly formed strand, or we can say leading strand is a newly formed continuous strand formed on the 3', 5' template strand. Okay, but the lagging strand is the fragments or discontinuous strand formed on the 5', 3' template strand. Is it clear? And this discontinuous fragments are called Okazaki fragments. Okazaki fragments are formed in the lagging strand. Okazaki fragments. Later, these fragments can be joined by the ligase enzyme. Okay, the ligase enzyme can join these fragments. Is it clear? I will show you. So, replication fork. Primase enzyme, right? You already learned. So, the DNA polymerase enzyme start polymerization. Next, we can see the video. Listen carefully.
The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called Primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA Polymerase binds to the primer and will make the new strand is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication.
Okay, so here the primer added by the primase can be removed by the exonuclease, then that gap can be filled by the DNA polymerase again. Hope you understood everything about the DNA replication. That's all for today.